In this lecture, we're going to discuss the various types of DNA polymorphisms and also the methods used to detect these polymorphisms in human DNA. At the end of this lecture, you should be able to compare and contrast the different types of polymorphisms, to define restriction fragment length polymorphisms, to describe short tandem repeat structure and nomenclature, describe genetic identification using the amylogenin locus, illustrate the use of short tandem repeats for bone marrow engraftment monitoring, define single nucleotide polymorphisms, and discuss mitochondrial DNA typing. A DNA polymorphism, we went over this a while back. This is a sequence difference compared to a reference standard, and the key is that this variation is present in at least 1 to 2% of the population. Polymorphisms can be single bases or thousands of bases, and they may or may not have phenotypic effects. So remember, that's what differs a polymorphism from a mutation. A mutation has a deleterious effect on the phenotype, whereas a polymorphism, it varies whether it's going to affect the phenotype. Polymorphisms are found throughout the entire genome. If the location of a polymorphic sequence is known, that sequence then serves as a landmark or a marker for locating other genes or genetic regions. So each polymorphic marker has different versions or alleles. It's estimated that genome sequences differ by one nucleotide every 1 to 1.5 kilobases. Single nucleotide polymorphisms, or SNPs, or what are called SNPs for short, may occur in both the coding and non-coding regions of the DNA. The human leukocyte antigen, or HLA locus, is a very highly polymorphic region in human DNA. So SNPs occur more frequently in the human HLA locus, and the variability in the HLA locus establishes self-identity of the immune system. So it's very important for determining compatibility between immune systems of transplant recipients and donors. The HLA locus is also used for exclusion in human identification cases. So types of polymorphic DNA sequences. Well, there are RFLPs, or restriction fragment length polymorphisms. There are VNTRs, or variable number tandem repeats, and these are 8 to greater than 50 base pair repeats. There are STRs, or short tandem repeats, which are 1 to 8 base pair repeats, and there are SNPs, which are single nucleotide polymorphisms. So one type of polymorphism we're going to discuss is a line, or a long interspersed nucleotide sequence. So lines are very highly repeated sequences that are between 6 and 8 kilobase pairs. They contain RNA polymerase promoters and open reading frames related to reverse transcriptase. There are more than 500,000 line elements, and lines make up more than 15% of the genome. There are also areas in the genome called signs, or short interspersed nucleotide sequences, and these are scattered throughout the genome. They are about 0.3 kilobase pairs in size. They're present in over 1 million copies per genome. So an example of a sign is an ALU element, and these are areas of the genome that have the ALU1 restriction site. So this is the recognition sequence for the ALU1 restriction enzyme, or restriction endonuclease. So both lines and signs are considered mobile or transposable elements. They can move throughout the genome. So they're copied and then spread by recombination and also reverse transcription. So these lines and signs may be responsible for forming pseudogenes, and pseudogenes are genes that don't have an intron and they are non-functional copies of active genes. Short tandem repeats and variable number tandem repeats. So those are two different types of repeat regions in the genome. These are shorter blocks of repeated sequences, and they both can undergo either expansion or shrinkage throughout generations. Restriction fragment length polymorphisms are polymorphisms that are detected through changes in the restriction map of a DNA region. So RFLPs are observed as differences in the sizes and number of fragments that are generated after restriction enzyme digestion of DNA and then southern blotting. So any nucleotide changes or polymorphisms have the potential to either remove, change, or create restriction endonuclease sites. And when these restriction enzyme sites are either added or removed, it's going to alter the number of fragments that you get after a southern blot when you digest with a specific restriction enzyme. So the first step in using your RFLPs is to construct a restriction enzyme map of the DNA region under investigation, so you know what the pattern, the banding pattern, should look like. So once the map is known, the number and sizes of the restriction fragments after restriction digestion of a test DNA region are compared with the banding pattern, sizes, and fragments of those that are expected. So RFLPs were initially done using the southern blot technique that we previously discussed. So the restriction fragment sizes are altered by changes in the enzyme recognition sites or in the region of DNA between restriction enzyme recognition sites. So here you see an example of double-stranded DNA, a small sequence, and you could see that there is a restriction site, GAATTC. So if you have a point mutation where you get GAAATC, you no longer have that particular restriction enzyme recognition site. You can also have an insertion or duplication, or you can have deletions and insertions in the DNA in between two restriction sites, and those are going to change the sizes that you would get when you run this on a gel and then blot. So the presence of your restriction fragment length polymorphism is inferred from changes in the fragment sizes. So for example, you have two different restriction sites in a particular genomic sequence. If a polymorphism were to occur, you would lose one of these restriction sites. So in an individual that had wild type sequence, they would have both restriction sites and they would have 3 bands on their gel that would be equal to in size, in this example, of the A fragment, the B fragment, and the C fragment. If an individual had the first restriction site, but there was a polymorphism where the second restriction site was lost, they would only have 2 bands on the gel. So the banding pattern, or what's called the fingerprint on the gel, is going to tell you if that individual has a polymorphism. So remember, in southern blot, you take your genomic DNA, you digest it, you run it on an agarose gel, you then blot the DNA onto a nitrocellulose membrane, you block the membrane, you then add your probe, and you want your probe to hybridize to a specific region of your DNA. You hybridize your probe, you wash off all the excess probe, and then you visualize your banding pattern. And depending on the method that you used, if it's radioactivity, you're going to do an auto-rad, you're going to use X-ray film. If you use chemiluminescence, that would be a colorimetric technique. So depending on which method you use, you're going to look at your bands in various ways. So for example, again, if you had the 2 restriction sites and your probe were specific to the DNA sequence within the region B of the DNA, then you would get a nice large band that was equal to fragment B. So depending on where this probe sits down, so if you lose the second restriction site, that probe is going to sit down on B, but since the B and C were not cut, you're going to have a much larger fragment where the probe sits down on B. So depending on the location of the fragment, you know if you had a restriction site or not. So restriction fragment length polymorphism genotypes are inherited. So for each locus, one allele is inherited from each parent. Each chromosome carries its polymorphisms so that the offspring inherits a combination of the parental polymorphisms. And due to recombination and random assortment, each person then has a unique set of restriction fragment length polymorphisms, half of which are going to be inherited by the mother and half of which are going to be inherited by the father. So over many, many generations, you get recombination events, mutations, a lot of other events that can alter the DNA sequence, which is going to increase the diversity of your DNA sequence. So a single locus, meaning a gene or a region of your DNA, will have several versions or alleles. And since humans are diploid, each person is going to have 2 alleles for each locus. If the alleles are the same or identical, then that locus is homozygous. And if the alleles are different, then the locus is heterozygous. So you can look at your RFLPs and you could determine, so you could look at the father at 2 different loci, you could look at the mother at your 2 loci, and then you could look at a child from the biological parents. At each locus, the child should inherit 1 from the mother and 1 allele from the father. And you should be able to see that for any locus that you look at when you look at the mother, the father, and a child. So not every single child in a family are going to inherit the same alleles. So each child is going to vary, but each child is going to inherit 1 from the mother and 1 from the father. So this is really important in identity testing and paternity testing and things like that. So parentage testing, if you look at an alleged father 1 and at 2 loci, and you look at alleged father 2 and the mother, and then you look at the child's RFLP at the same 2 loci, you should be able to determine which of the alleged fathers is the actual biological father. So let's look at the child. The child in locus 1 has the green band from the mother and a black band from the father. So you could see that at locus 1, the black band is the same size as one of the bands in locus 1 from alleged father 2. When you look at locus 2, you could see the green band from the mother, so the other band is from the father, and once again, that band is not present at that size in locus 2 of alleged father 1. So alleged father 2 is the actual biological father of this child. So you can also do evidence testing using restriction fragment length polymorphism. So you can look at various suspects, you could look at the victim, you could look at crime scene evidence, and you could compare those sizes to your molecular weight standard. So in this instance, you have 3 different loci, you have 2 different suspects, and you want to know which one was at the crime scene. So you see your banding pattern of suspect 1 at locus 1, suspect 2 at locus 1. You could see the victim's pattern, and you can see the crime scene evidence. So if you look at locus 1, the crime scene evidence matches the victim and suspect 2. So in locus 1, suspect 2 would be the person that may have been at the crime scene. If you look at locus 2, you have suspect 1, suspect 2, the victim, and the evidence from the crime scene. Once again, you have the victim's banding pattern and you have suspect 2's banding pattern. And then you do the same thing for locus 3. So at all 3 loci, you have the band that matches the victim and you have the band that matches suspect 2 from the evidence of the crime scene, which implicates suspect 2 for being present at the crime scene. You can use RFLP for human identification. So what you have in genomic DNA are tandem repeats of sequences of all different sizes. So your repeats of 8 nucleotides or more are called variable number tandem repeats or mini satellites. The repeats are large enough where if you have a loss or a gain of 1 repeat, you can see this when you resolve your bands on a gel after you use your restriction digestion, your restriction enzymes. Many times when you want to do this type of banding pattern or restriction fragment length polymorphism, you're going to use restriction enzymes that are frequent cutters, meaning that they cut very often in a DNA sequence. They recognize very small nucleotide sequences, only 4 or 5 nucleotides that are present very commonly throughout the genome. So they're going to cut a lot. So examples of frequent cutters are HAE3 or HINF1. And these are going to cut frequently and generate very small fragments. So if you have a loss or gain of a repeat, you're going to be able to see that variation in the banding pattern. You can do something called multiple locus probe RFLP, where you can use 3 to 5 probes to analyze 3 to 5 different loci on the same blot. So using multiple probes at the same time to produce patterns gives you high variability between individuals. However, using these multiple probes can be very difficult to analyze and interpret. So since the multi-locus probes are difficult to analyze, there was a development of a single locus probe RFLP where you analyze 1 locus at a time. So you get a much simpler pattern. It's easier to interpret. However, it does require computer analysis to accurately size those restriction fragments. And if you get a match, that would imply that you can include, or this is an inclusion criteria for two people to be the same. So the probability of two people having the same set of restriction fragment length polymorphisms or a profile becomes lower as more loci are analyzed. So you're analyzing one locus at a time using a single probe. And if you analyze numerous loci with different probes and then compare each one of those profiles, you're going to get a lower probability of two people having an exact same pattern or profile. Short tandem repeats are also known as microsatellites. Now, short tandem repeats are very similar to variable number tandem repeats or mini satellites. However, short tandem repeats have smaller repeat units of 1 to 7 base pairs. So sometimes short tandem repeats contain repeat units with altered sequences. These are called microvariants. So the repeat unit might be missing one or more bases of that repeat sequence. Short tandem repeats are efficiently amplified using polymerase chain reaction, and since you can amplify the short tandem repeats, it requires a lower amount of the specimen. Also, using PCR technology is much faster than southern blotting. Southern blotting can take 3 to 4 days. PCR takes 3 to 4 hours. So this is a nice, efficient, rapid technique. So here are some examples of short tandem repeat polymorphisms. So short tandem repeats are repeats of a nucleotide sequence. For example, you could have a repeat of one nucleotide, A. That's a mononucleotide repeat. You can have a dinucleotide repeat, AT. Trinucleotide, 3 nucleotides repeated over and over, TAG. Tetranucleotide, pentanucleotide. So these are examples of short tandem repeats. Different alleles are going to contain different numbers of repeats. So for example, here you have one allele that has a TTC repeat 4 times. But the other allele has the TTC repeat 5 times. So you can analyze your short tandem repeat alleles by fragment size using a southern blot technique. So for example, you have your allele 1 and you have an AGCT repeat. And in allele 1, that repeat, AGCT, you have that repeat 4 different times. That repeat is in between a specific 4 base pair restriction enzyme recognition site, GGCC. So whenever the restriction enzyme that recognizes GGCC sees that 4 base pair sequence, it is going to cut. So for allele 2, you have the same recognition sequence, GGCC, and you also have a tandem repeat in between the recognition sequence. Now in the first allele, allele 1, we have 4 repeats. In the second allele, we only have 3 repeats. So if you cut your DNA with this restriction enzyme and use a probe that recognizes that repeat or that area and hybridizes, you're going to get a larger fragment with allele 1 than you will with allele 2 because allele 1 has 1 more repeat. So it's going to be a larger piece of DNA. Short tandem repeat alleles can also be analyzed by your amplicon size through polymerase chain reaction. So what you would do is you would make your primers that are homologous to your DNA sequence outside of your short tandem repeat. you would run your PCR reaction and then run your band on a gel. So for example, for allele 1, you have 7 repeats, so you're going to get an amplicon size of 187 base pairs. But in allele 2, you have 8 repeats, so the extra repeat is going to make your amplicon slightly larger, so 8 repeats, you're going to get a 191 base pair fragment. So in this instance, the genotype is 7-8 because allele 1 has 7 repeats and allele 2 has 8 repeats. You can use allelic ladders to standardize your short tandem repeat polymorphism. So these allelic ladders are standards that represent all of the alleles that are observed in a population. So for example, in this allelic ladder, the bottom band indicates 5 repeats and the top band indicates 11 repeats. If you run an individual on a gel and compare it to the standard allelic ladder, you can see that the second lane, that individual has a genotype of 7-9 because they have a banding pattern of 7 repeats and 9 repeats. The second individual has a genotype of 6-8 because they have 6 repeats and 8 repeats. So multiple loci are genotyped in the same reaction using your multiplex PCR. And remember, multiplex PCR uses several primers, and you can amplify various regions in a DNA sequence at the same time. So instead of setting up 2 or 3 separate PCR reactions, you set up your reaction all in the same tube using various primers. So the thing about your multiplex PCR, your allelic ladders can't overlap in the same reaction, or you won't be able to see your various bands. So here's your short tandem repeat polymorphism using multiplex PCR. So for example, in the picture on the left, you have 4 different loci that are being analyzed. So you're going to have 4 different sets of primers in that example. On the right, there are 5 different loci, so 5 different sets of primers. So you can look at the FGA locus, the TPOX locus, and see the genotype at each locus. So the nomenclature for your short tandem repeats is generated by the International Society for Forensic Genetics. So your short tandem repeats within genes are designated according to the gene name. So THO1 is in intron 1 of the human tyrosine hydroxylase gene on chromosome 11. TPOX is in intron 10 of the human thyroid peroxidase gene on chromosome 2. So these particular short tandem repeats don't have a phenotypic effect within these genes. You also have short tandem repeats that are not associated with genes. And if they are not associated with a gene, they have a standard designation, which is D a number, S a number. So D is the DNA. The first number is the chromosome where that short tandem repeat is located. The S is a unique segment, and the last number is the registration number in the International Genome Database. So you can use short tandem repeats for gender identification, and this is commonly done using the amylogenin locus. So amylogenin is not a short tandem repeat. So the amylogenin gene is located on the X and Y chromosomes, and it's required for embryonic development and tooth maturation. The polymorphism is located in the second intron of the gene. The Y allele of the amylogenin gene is 6 base pairs larger in this region than compared to the X allele. So amplification and resolution reveals 2 bands or peaks depending on if you're running on a gel or using an electropherogram. So you're going to get either 2 bands or 2 peaks for a male, which is going to indicate XY, and you're going to get 1 band or 1 peak in a female because it's XX. So the amylogenin locus, you're using the HUMAMEL gene that codes for amylogenin-like protein. So the gene is located at XP 22.1 to 22.3 and Y. So on the X allele, you're going to get a 212 base pair band. On the Y allele, you're going to get a 218 base pair band because it's 6 base pairs larger. So in analyzing your test results, the discovery of the same set of alleles from different sources or shared alleles between related individuals is very strong evidence of identity, paternity, or relatedness, and it's expressed in terms of the background probability of chance matches. So DNA testing results in your peak or band patterns are converted to a genotype or allele identification, so you can compare your results between labs. Because if one lab runs a gel and another lab runs a gel, it's very difficult to determine that both 2 bands are absolutely the same. So if you convert your bands into a genotype compared to a standard, then you can 2 different labs can say, well, I got genotype 6-8, and it's much easier to compare the results. So a short tandem repeat locus genotype is defined by the number or repeats in the alleles. So if the locus genotype is heterozygous with 7 repeats on one chromosome and 8 on the other, the locus is designated 7/8 or 7,8. A homozygous locus is designated by the single number of repeats of that allele, 7-7, 7,7, or just 7. Microvariant alleles containing partial repeat units are indicated by the number of complete repeats, followed by a decimal point, and then the number of bases in the partial repeat. So for example, 9.3. So what you're generating when you determine these genotypes is your profile. So the genotype or the profile of a specimen is the collection of alleles in all of the locus genotypes tested. A matching genotype is not necessarily an absolute determination of the identity of an individual. Genetic concordance is used to express the situation where all locus genotypes or alleles from 2 sources are the same. So concordance is interpreted as inclusion of a single individual as the donor of both genotypes. 2 samples are considered different if at least one locus genotype differs, and this would be exclusion. There's one exception though. In paternity testing, you might have a mutational event that could generate a new allele in the offspring. So then you're going to get a difference in one locus. So in that case, you may not be able to exclude. So once you get your profile of different specimens, you're then going to match your profile. So your results are going to be used to determine the probability of identity or inheritance of genetic markers, or to match a particular marker or marker pattern. So the chance that the same allele could arise in the population randomly must be taken into consideration. So the frequency of a set of alleles or a genotype in a population is the product of the frequency of each allele separately, and this is known as the product rule. So linkage equilibrium assumes that the loci are not associated with one another, or what's called genetically linked in the genome. The overall frequency, or OF, of a locus genotype consisting of n, or the number of loci, can be calculated. So you can take your overall frequency, and that is going to be calculated by the frequency of each loci, multiplying those together. So F is the frequency of each individual allele in the population. And the individual allele frequencies are determined by testing many individuals in general and defined populations. For example, at locus penta D on chromosome 21, the 5 allele has been determined previously to occur in 1 out of 10 people. At locus D7S829 on chromosome 7, the 8 allele has been observed in 1 out of 50 people in the same population. So the overall frequency would be 1 over 10 times 1 over 50, which equals 1 over 500. This means that this genotype or profile would be expected to occur in 1 out of 500 randomly chosen members of that population. So the more loci that you test, the greater the certainty that the profile is unique to a single individual in that population. So here's an example of analysis of short tandem repeat polymorphisms using PCR. So your short tandem repeat genotypes are analyzed using either gel or capillary gel electrophoresis. For example, you would run your PCR on a gel and compare it to your allele ladder, and you would see the number of repeats that you get. You can also do this in your capillary gel electrophoresis, where you get your peaks, and you're going to compare the peaks that you get to your allelic ladder and determine your genotype. So your short tandem genotypes are inherited, so you can use your PCR short tandem repeat analysis to compare a mother, a father, and a child to determine paternity. So doing your parentage testing by short tandem repeat PCR, you can look at various loci between a child, a mother, and 2 alleged fathers. And then you could determine the genotype at each locus. For example, at D3S1358, the child's genotype is a 1617. The mother is a 16, and the alleged father, 1 is 17 and 2 is 17. So that locus, you wouldn't be able to decipher which alleged father might be the biological father, because they have the same genotype at that locus. For the locus VWA, the child is 1418 and the mother is 1618. The alleged father 1 is 1415 and alleged father 2 is 1617. So since the mother has the 18, the child must have gotten the 18 from the mother. So that leaves the 14 from the father, and only alleged father 1 has the 14. However, just looking at this one locus isn't enough to say that number 1 is the biological father. You would have to look at numerous loci, because the more loci you look at, that's going to lower the probability that some person randomly chosen would have the same genotype. So when you look at the FGA locus, again, both fathers have the same genotype, so you wouldn't be able to really use that locus. At the TH01 locus, the child is 6 or 6'6", the mother is a 6 and a 9.3. Notice that the mother has a microvariant there. The alleged father's a 1'6" and a 6'7". So the child is a 6'6", so it got the 6 from the mother and a 6 from the father. Both fathers, alleged fathers, have a 6. TPOX, child is a 10'11", mother is a 10'11". So father 8'11" and 8'9". Well, since the child is a 10'11", it obviously got the 10 from the mother and the 11 from the alleged father 1. So you would have to look at all of these different loci and see if one of these potential fathers is a match with the child, comparing the mother and the child's genotypes. And after looking at all of these loci, you can determine that the probability of that father being anyone else besides this particular individual is very, very low. So again, evidence testing by short tandem repeat PCR. Which suspect was at the crime scene? You have two different suspects. You have your victim, you have your crime scene evidence, and you have your allelic ladder. So when you look at suspect 1, suspect 2, the victim, and the crime scene evidence, the crime scene evidence is going to match the victim and suspect 1 in each locus, and after numerous loci you can rule out anyone else but suspect 1 as being the most likely suspect. You can use Y-STRs to look at short tandem repeat polymorphism. So the Y chromosome is inherited as a block. So the Y chromosome does not undergo recombination. So short tandem repeats on the Y chromosome are inherited paternally as a haplotype. Y haplotypes are used for exclusion and paternal lineage analysis. So except for very rare mutation events, every male member of a family is going to have the same Y chromosome haplotype. You can use short tandem repeats for chimerism testing. So for example, in bone marrow transplants, the transplants are monitored using short tandem repeats. So you can have an autologous transplant or a transplant from the same individual. So the donor and recipient are the same person. So the recipient is going to receive his or her own cells where the cancer cells have been removed. In an allogenic transplant, you have a different donor and recipient. So the recipient is going to receive cells from the donor. So the recipient with the donor marrow is considered a chimera because they have their own cells as well as the donor cells. So there are two parts to chimerism testing using short tandem repeats. You have your pre-transplant informative analysis. So you want to determine what's the genotype of the donor and what's the genotype of the recipient before transplanting the cells. Then you want to do engraftment analysis after the transplant to see what the genotype is. If after the transplant the genotype matches the donor completely, that would be considered complete or full chimerism. If you see mixed cells, both donor and recipient, that would be considered a mixed chimerism. And if you only see recipient genotype and not donor, that would be considered a graft failure. So the recipient did not accept the donor cells. So in the informative analysis, your short tandem repeats are scanned to find informative loci. So what you want to do is find donor alleles that differ from the recipient alleles. So those are considered your informative loci. You wouldn't want to find alleles from the donor and the recipient that are identical, because then you're not going to be able to determine after the transplant what's the donor and what's the recipient. So for example, if you look at 5 different loci, you have your marker, your donor, and your recipient, which loci in this example would be considered informative? So for locus 1, your donor and your recipient genotype is exactly the same, so that's not going to be informative. Locus 2, your donor and recipient genotypes are completely different, so that is an informative locus. Locus 3, the recipient and the donor have some similarity, but the donor does have a difference, so that could be informative. Locus 4, not informative at all, they're identical. Locus 5, that would be informative because the donor and recipient genotypes are different. So there's different degrees of informativity. The most informative loci, your recipient bands or peaks don't overlap or stutter in the donor bands or peaks. So stutter is a technical artifact of a PCR reaction where a minor product of your N-1 repeat unit is produced. So for example, you have your informative loci, you have your recipient peak pattern, you have your donor peak pattern. And you could see in the donor peak pattern in the second example down from the top, you could see a stutter is generated. You have another locus, you have your recipient and your donor. And then examples of non-informative loci. So for this locus, the recipient and donor have the exact same peaks in the same sizes and same locations. So that's going to be a non-informative locus. So which loci are informative in this pattern? So you want to see where do you have different peaks, different sizes and different locations of your peaks. So all of these loci would be considered informative. So you can use your chimerism testing for engraftment analysis. So using those informative loci, your peak areas are determined in fluorescence units because each peak is a fluorescence. You can also scan your gels and do densitometry. So how dense are the bands on your gel? So the AR is the area under the recipient specific peaks, and the AD is the area under the donor specific peaks. And there's a formula to calculate the percent recipient or percent donor. So the percent recipient DNA is the AR over the AR plus the AD times 100, and the percent donor DNA is the AD over the AR plus AD times 100. So here's an example to calculate the percent recipient DNA in the post 1 and post 2. So you have recipient at the locus D16S539, and you have the donor. You have the recipient's whole blood after transplant, and you have the recipient's T cell fraction. So the area under the peaks is used to calculate the percentages. These are your fluorescence units, and you could see the number underneath the post peaks. So in order to calculate the percent recipient DNA, you have your R unshared over your R unshared plus D unshared. So that is going to be your 4,616 divided by 4,616 plus 16,413 times 100, and that's going to mean that there are 22% recipient cells in the unfractionated blood. And if you also do the 516 divided by 516 plus 15,608 times 100 in the T cell fraction, you determine that there are 3.3% recipient cells in that T cell fraction. So in order to analyze your cellular subsets for chimerism, your T cells, granulocytes, NK or natural killer cells, all your different types of cell subsets, engraft with different kinetics. So analysis of your cellular subsets provides a more detailed description of the engrafting cell population. It also increases the sensitivity of the engraftment assay. So if you look at your cellular subsets, so your T cells, maybe your CD3 T cells, your CD56 NK cells, your CD13 and CD33 myeloid cells, etc. So you can look at the markers on your cells using flow cytometry sorting or FACS, immunomagnetic cell sorting, as well as immunohistochemistry and XY fluorescent in situ hybridization. So you can detect your different levels of engraftment in your cellular subsets in a split chimerism. So for example, you have your recipient or R, your donor D, your T is your T cell subset, and in this example it's mostly recipient cells, and your G, your granulocyte subsets, are mostly donor cells. Single nucleotide polymorphisms are single nucleotide differences between DNA sequences. So one SNP occurs approximately every 1,250 base pairs in human DNA. SNPs are detected through sequencing, melt curve analysis, and various other molecular methodologies. 99% of SNPs have no biological effect, and 60,000 SNPs are within genes. So you can do your SNP detection through sequencing, and remember, your sequencing is determining the exact nucleotide sequence in a region, and it's done through capillary electrophoresis, looking at fluorescence, and you can look at your electropherogram to see what nucleotide you have at that location and compare it to the wild type sequence. SNP haplotypes, so your SNPs are inherited in blocks or haplotypes, so the entire block is going to be inherited to the offspring. So SNPs can be used for mapping genes, for human identification, chimerism analysis, and numerous other applications. The Human Haplotype Mapping, or HapMap project, is aimed at identifying single nucleotide polymorphism haplotypes throughout the entire human genome. You can also look at mitochondria to identify DNA polymorphisms. So in mitochondria, there are what's called hypervariable regions. And in mitochondria, there are specifically two regions that are considered hypervariable, HV1 and HV2. So your mitochondria is a circular genome that's inherited maternally. there's an average of 8.5 base differences in the mitochondrial hypervariable sequences of unrelated individuals. All maternal relatives will have the same mitochondrial sequences, and mitochondrial typing can be used for legal exclusion of individuals or confirmation of maternal lineage. So in summary, there are 4 types of polymorphisms used for various purposes in the laboratory, and these specifically are RFLP, VNTR, STR, and SNPs. Polymorphisms are used for human identification and parentage testing. Y-STR haplotypes are paternally inherited. Polymorphisms are used to measure engraftment after allogenic bone marrow transplants. SNPs are detected by sequencing, milk curve analysis, or other methods. SNPs can be used for the same applications as other polymorphisms. Mitochondrial DNA typing is performed by sequencing the mitochondrial hypervariable regions, and mitochondrial DNA is maternally inherited.